In this video, I will make an overview of translation. When you take a moment to look at your hands, the bone, skin, and muscle you see are made up of cells, and each of those cells contains many millions of proteins. As a matter of fact, proteins are key molecular building blocks for every organism on Earth. How are these proteins made in the cell? For starters, the instructions for making proteins are written in a cell's DNA in the form of genes. If that idea is new to you, you may want to check the section on DNA and RNA before getting into this building proteins in instruction. So basically, a gene is used to build a protein in two-step process. The step one, transcription. Here the DNA sequence of a gene is rewritten in the form of RNA. In eukaryotes like you and me, the RNA is processed and often has few bits snipped out of it to make the final product called messenger RNA, or mRNA. In step 2, translation, in this stage, the mRNA is decoded to build a protein or a chunk, subunit of a protein that contains a specific series of amino acids. So, I will try to zoom in on translation in this video, getting an overview of the process and the molecules that carry it out. Okay, so the genetic code. During translation, a cell reads the information in a messenger RNA or mRNA and uses it to build a protein. Actually, to be a little more technical, an mRNA doesn't always provide instructions for a whole protein. Instead, what we can confidentially say is that it always encodes a polypeptide or a chain of amino acids. In an mRNA, the instructions for building a polypeptide are RNA nucleotides, A's, U's, C's, and G's, read in groups of 3. These groups of 3 are called codons. And there are 61 codons for amino acids, and each of them is read to specify a certain amino acid out of the 20 commonly found in proteins. One codon, AUG, adenine, uracil, and guanine, specifies the amino acid methionine and also acts as a start codon to signal the start of a protein construction. There are also 3 more codons that do not specify amino acids. These stop codons, UAA, UAG, and UGA, or uracil adenine adenine, uracil adenine guanine, and uracil guanine adenine, that tell this cell when a polypeptide is complete. So they are the stop codons. All together, this collection of codon amino acid relationships is called the genetic code, because it lets cells decode an mRNA into a chain of amino acids. Okay. So, transfer RNAs, maybe you have heard of tRNAs. Transfer RNAs or tRNAs are molecular bridges that connect mRNA codons to the amino acids they encode. one end of each tRNA has a sequence of 3 nucleotides called an anticodon, which can bind to specific mRNA codons. The other end of the tRNA carries the amino acid specified by the codons. There are many different types of tRNAs. Each type reads one or a few codons and brings the right amino acid matching those codons. So ribosomes are, what you see in the picture is ribosomes. Ribosomes are the structures where polypeptides or proteins are built. They are made up of protein and RNA, ribosomal RNA or RNA, rRNA. Each ribosome has 2 subunits, a large one and a small one, which come together around an mRNA, kind of like the 2 halves of a hamburger or bun coming together around the patty. The ribosome provides a set of handy slots where tRNAs can find their matching codons on the mRNA template and deliver their amino acids. These slots are called the A, P, and E sites. Not only that, but the ribosome also acts as an enzyme, catalyzing the chemical reaction that links amino acids together to make a chain. So when it comes to the steps of translation, your cells are making new proteins every second of the day, and each of those proteins must contain the right set of amino acids linked together in just the right order. That may sound like a challenging task, but luckily your cells, along with those of the other animals, plants, and bacteria, are up to the job. To see how cells make proteins, let's divide translation into 3 stages. Initiation, starting off, elongation, adding onto the protein chain, and termination, the finishing up. So getting started in the initiation, the ribosome assembles around the mRNA to be read, and the first tRNA carrying the amino acid methionine, which matches the start codon AUG. This setup, called the initiation complex, is needed in order for translation to get started. Next, elongation, extending the chain. You see in the picture. Elongation is the stage where the amino acid chain gets longer. In elongation, the mRNA is read 1 codon at a time, and the amino acid matching each codon is added to a growing protein chain. Each time a new codon is exposed, a matching tRNA binds to the codon. The existing amino acid chain polypeptide is linked onto the amino acid of the tRNA via a chemical reaction. The mRNA is shifted 1 codon over in the ribosome, exposing a new codon for reading. During elongation, tRNAs move through A, P, and E sides of the ribosome, as shown in the picture. This process repeats many times as new codons are read and new amino acids are added to the chain. So when it comes to finishing up, which is the termination, termination is the stage in which the finished polypeptide chain is released. It begins when a stop codon, UAG, UAA, or UGA, enters the ribosome, triggering a series of events that separate the chain from its tRNA and allow it to drift out of the ribosome. After termination, the polypeptide may still need to fold into the right 3D shape, undergo processing such as the removal of amino acids get shipped to the right place in the cell or combine with other polypeptides before it can do its job as a functional protein.